Research into eastern brown snakes has found that they have different venom depending on whether they're from the north or the south of Australia. That raises questions about whether antivenom is as effective as it could be. Professor Bryan Fry is from the University of Queensland and he joins us now. Hi there, Bryan. Thanks for joining us.
Thank you for having me.
So tell us then a little more about the different types of venom produced by brown snakes.
Well, what we found was a real surprise where we set out to look at brown snakes from across the range and all the different species and multiple populations within each species. And to our surprise, we found that only the brown snakes in the southern half of Australia have the venom like what we expected to find of this taipan-like venom, but where they form strong, robust blood clots. But what we found for all other populations of the eastern brown snake and all other species of brown snake is that they make these tissue-like blood clots that break down almost immediately. And this may go a long way to explaining the strange phenomenon that's quite unique about brown snakes, where they're the only snakes in the world that can kill within 15 minutes regularly. And for the first 13 minutes, the patient's actually feeling fine. And it's those last 2 minutes that are just a catastrophic decline, where it's like falling off a cliff. And with this change in the venom between the different populations, we think that the ones that are having this strange effect on the blood may be the ones that are causing the cardiovascular collapse. Right. But we don't know this for any certainty looking at the clinical records, because all the amalgamation of clinical records of brown snakes have historically just taken hundreds of cases, lumped them together. So it hasn't been stratified geographically. So we're going to go back and wade through these hundreds of cases. And in the southern population of the eastern brown snake, it's the only brown snake that lives in that area, versus other parts of Australia, you'll have 2 or 3 species of brown snake living alongside of each other. So we can partition it geographically to that temperate zone versus everything else, and then start seeing are there clinical differences that match the venom differences.
Okay, but there's only one antivenom, isn't there, that's given to all brown snake bites, irrespective of where the snakes are from?
Yeah, and that's the big question, because it's made just using the eastern brown snake venom. We've, of course, up until this study, no one knew that there's a difference in the venom between different populations. So CSL gets their venom from the Australian Reptile Park, who has brown snakes from New South Wales as well as Queensland. Now, the snakes, the southern snakes you find on like coastal New South Wales through ACT, Victoria, and South Australia are a much smaller type of brown snake than the Queensland population of the same species. They get a lot bigger. So they're going to get more venom from a Queensland snake than a temperate zone snake. So the venom pool may be biased towards one and not include much of the other, which we want to emphasize that the antivenom still works, but it might just require proportionally more. But what this means is that really it should be balanced between the 2 venom phenotypes. That there should be 50% of the antivenom is made using or 50% of the venom that goes into the antivenom production is from the temperate zone snakes, and 50% is from the more tropical snakes, so that we get an even distribution of the types of antibodies and therefore get more complete coverage. But first, we're going to do tests with multiple batches of antivenom and see, is there a difference and how well it performs for northern populations of the eastern brown and southern populations of the eastern brown, and also all the other species of brown snake. So this is that classic kind of study where it's a hydra. We answer one question, now we have 7 more interesting ones that we're going to spend a few years pursuing.
And just finally, and briefly if you could, Bryan, what's the reason for the difference between the venom of the southern and the northern brown snake? What's behind it?
That's a really good question, and that's another one of the new questions that we need to answer. That it's fascinating that it's only that temperate population that has that taipan-like venom. All the other populations of the eastern brown and all other species of brown snake have this unique one. So we're seeing a stratification based on temperature. So does this mean different prey type, different prey escape potential? We don't know, but that's going to be something that'll be a lot of fun field work to answer.